We're starting on chapter 6, talking about cells, cell theory, organelles, and microscopes. Yes. All right, so this is all in chapter 6. We'll soon be doing chapter 7, which is cell membranes as well. So here's some of the page numbers, 94 to 108 and 108 to 124 to kind of break up that reading for chapter 6. Remember, the different types of cells include, well, from the kingdoms, animal cells, plant cells, fungus cells, protist cells, and then the two types of bacterial cells that belong to archaea and eubacteria. But if we can even categorize those further into the types of domains, and organisms that are in the domains of, that are in the kingdoms of animal, plant, fungus, and protist all have the eukaryotic cell and are in the eukarya domain based on that type of cell. It means they have the complex nucleus inside and the other organelles that are membrane bound that have specific jobs. The other domains, archaea and eubacteria, are prokaryotes. They have the prokaryote type of cell where there is no nucleus, no complex specific job organelles, and the DNA is just floating around in the cytoplasm there. So that means all those jobs that those organelles do, those specific jobs, that means in the bacteria, they're done either in the cytoplasm by molecules or by molecules like proteins that are embedded in the membrane of the cell of the prokaryote itself. Look at the different sizes. Wow. If a eukaryote cell was the size of a watermelon, the bacteria cell would be like a watermelon seed. And we are multi-celled organisms. So lots of cells there, but they get together to build tissues that do certain jobs, and then those tissues get together to build organs, and then the bodies or organ systems. And so it's part of that hierarchy theme that we talked about in chapter 1. Remember, cells do all the work of life. They're so busy. All these different things like breathing and taking in energy or taking in food to make energy, building molecules, removing wastes, maintaining homeostasis, and building more cells. Such a busy everyday life that they have. If I again had to pick my 3 favorite and main jobs that cells do, making energy in the form of ATP, all cells have to do that. Even plant cells have to do it. It's just that plant cells make their own glucose first, and then they use that glucose to make their ATP. Whereas we have to eat our glucose first and then make our ATP. They all make proteins. Proteins are those multifunctional molecules that get so many jobs done. And so you've got to make those proteins. And remember, DNA is the recipe to make those proteins. And then making more cells, like mitosis and meiosis. I hope you remember those words for cell reproduction. You have to, since you're a multi-celled organism, you have to replace your cells. And then when you reproduce, you have to go through meiosis to make gametes or sperm and egg. Organelle. An organelle means a little organ. It has a specific job to do usually, sometimes more than one job. And its structure determines its function often, and it's surrounded by a membrane to keep it self-contained from the cytoplasm, or what's called the cytosol. That's the liquid part around it. All right, cells need power. So making energy, that's a big job, fuels the everyday life, the growth. You need to take in food and digest it. Cells also need oxygen in order to do this. They need their ATP, plus all the buildup of waste when the job is done. You've got to get rid of that. So organelles that do this work include the cell membrane, lysosomes, kind of like the cleanup crew basically, vacuoles and vesicles, those are good storage areas, and the mitochondria who make the ATP. So there's our cell membrane. So what we learned back in chapter 5 is going to really help now with those macromolecules, because here's one looking at us right now. A phospholipid, yeah, with the phosphate head giving it a charge, making it polar, and then the nonpolar or hydrophobic tail with the two fatty acid chains. And so those point inward, remember, to get away from the water that's in your bodies. And then the phosphate heads point outward because they don't care about the water that's in your bodies. So that helps to separate what's inside the cell from what's outside. And it controls what leaves and what enters, letting in the good stuff, getting rid of the bad stuff, and then recognizes signals from other cells so that you can do certain functions and make certain changes. The structure of a phospholipid bilayer is said in the name, 2 layers. The bilayer, bi means 2. So you've got all of those in their 2 layers with the phospholipids and then proteins embedded in there to allow things to come in and out, acting like carriers or transporters, or acting like receptors to receive information or to signal other cells. Let's take a look at an animal cell here. The cytoplasm is an inclusion of both the jelly-like material and the organelles themselves. So cytoplasm isn't just the liquid part of the cell, it also includes the organelles. And just the liquid is called the cytosol. And there's the cell membrane acting as the boundary around the cell. Now, vacuoles and vesicles, they move material around. And often, if we look at, this is an ER, we haven't talked about that yet, an endoplasmic reticulum. They're basically bundles of membrane material that act as little tunnels to pass things through. But they're really good at bubbling off and then making a vesicle like this to transport something, like maybe making a polypeptide and then transporting it to the Golgi apparatus, which is a little bit further out from the nucleus, more closer to the membrane. Functions to bring that polypeptide to the Golgi to finish that protein building. Golgis are very much like the ER, with membranous like tunnels, and really, really good at vesicling off, bubbling off, I guess maybe that's a better word, to transport things and even to make what are called lysosomes. If you've got your proteins, you made your proteins that are going to act as digestive enzymes and you bubble off, now you've got what's called a lysosome, a little vesicle that has digestive enzymes inside. And then that lysosome can merge with another vesicle that has trapped an old dead mitochondria and digest it. So kind of like the housekeeper, keeping things nice and clean in your cytoplasm. And here we go on the cell membrane. We've got what's called phagocytosis happening here, where there's a piece of food and it gets received by receptors here on the cell membrane, and the cell membrane bubbles inward to let that in, and then puts a membrane around it, which then that would be a vesicle filled with food. Now merge that with a lysosome with digestive enzymes, and bam, you've got a digestive vesicle or vacuole. And you can digest that food, let it absorb and diffuse into the cytoplasm, and that cell can be considered fed. If you're a one-celled creature, like a protist, that would be how you might eat. All right, so some food and water storage. Take a look at a plant cell, very prominent. A big, huge central vacuole where they can store things. Like when they make sugar, that's a great place to store all that sugar that they make for a rainy day for when they need to make ATP. For this protist here, this is a paramecium, lives usually in a freshwater situation, and well, water usually diffuses into their cell because of a concentration difference that we'll talk about a little bit later. And so to get rid of excess water that diffuses into their cell, they collect it into a vacuole. And once that vacuole gets filled up with water, it merges with the cell membrane. Cell membrane phospholipids are good at merging with each other, and then they can dump out that excess water. All right, so we added vacuole and vesicles to our cell picture here for transport and storage. What are we going to add next? Oh, lysosomes. Again, there's our lysosomes that I just talked about. The housekeepers of the cell. Usually they bubble off of the Golgi apparatus because the Golgi is good at doing that. And then the proteins got finished in the Golgi, so you've got some digestive enzymes that are proteins, bubble them off, and bam, you've got a lysosome. And you can digest different things, whether it be food or old cell parts that way. Here's an interesting job for lysosomes, because in your early development, your fingers and your toes actually have webbing in between them. That's just a trait left over from our ancestors who actually do have webbed feet and webbed fingers. But our lysosomes are supposed to release digestive enzymes to separate those fingers and toes, but sometimes that just doesn't happen. You probably know someone who has webbed toes. Maybe, maybe not. So there's a lysosome, that's the red one, for food digestion and, the garbage disposal and recycling. I like to think of it as the housekeeper of the cell. It has that prefix lyso, which is like Lysol. It's a cleaner. Lyso actually means destruction, if you haven't gathered that yet. Mitochondria. Everybody knows the mitochondria as the powerhouse of the cell. Its job is to make ATP. Actually, its job is to recycle ATP. After you use ATP, you remove the third P from the adenosine triphosphate. So mitochondria's job is actually to put that P back on to make the ATP reusable again. And it takes sugar and glucose to do that, or other macromolecules, but glucose is fastest. And the structure determines the function here, definitely. Take a look at a mitochondria. This picture on the right is using an electron microscope. You can see how the membrane actually folds partway in and comes back out. So you have all this surface area inside, and that's because there's different enzymes involved with making ATP that are embedded on all those infolded membrane parts. And so there's more surface area, more enzymes means more ATP. This is in both plant and animal cells. So the powerhouse of the cell, mitochondria, kind of look like little kidney beans, and there they are pictured in our cell. What's next? Oh, plants make energy two ways, with making ATP with the mitochondria, and then they have to make sugar from sunlight and carbon dioxide, of course. And so that's the process of photosynthesis. There's those equations there. We could add water into this as well. And the difference between our plant and animal cells there. Look at a plant cell or other producer cell, you're going to see a lot of green. That's due to the chloroplast, which is an organelle that they have that we do not have. That includes the green pigment chlorophyll. And the plant cell has the big central vacuole as well. But you'll notice both of them have the mitochondria. So now we switch to a picture of the plant cell, labeling the cytoplasm, the mitochondria, the cell membrane, the lysosome. Here's the chloroplast, the central vacuole, and animal cells do not have this, but plant cells have the extra covering called the cell wall. Now remember, cell walls in plants are made out of the polysaccharide cellulose, which we do not have an enzyme to digest because it's made with that beta glucose, the isomer of glucose, that creates the weird bonds that, well, we just don't have an enzyme for. All right, cells need workers, and those workers are proteins. So that's a regular ritual of daily life, reading those genes on DNA and building those proteins. So the organelles that do this work, we've got the nucleus listed, ribosomes, which actually technically are not considered organelles, and then the ER, endoplasmic reticulum, and the Golgi apparatus. So proteins do all the work. Why, yeah, they're like the workhorse of the cell. Remember all their different jobs, building structures, enzymes that do specific jobs or chemical reactions, acting as signals, receptors, acting as defense and storage proteins, helping other proteins get their right 3D shape, like those chaperonin proteins I showed you. So many different jobs that they have to do. So let's go to the nucleus. This is where the actual recipe lies for you to make your correct proteins. It's like the control center of the cell because it houses that DNA. The structure includes a nuclear membrane. You'll notice little like pores or holes. That's because things are made in the nucleus and actually will need to get out to the rest of the cell. And the nucleolus, it's like a nucleus of the nucleus. That's where ribosomes are made. Remember we talked about ribosomes back here. They are part of the protein making machinery. And then chromosomes, which are basically proteins that have DNA wrapped around them so that you have a place to store that DNA in a very organized fashion so that when it comes time to being able to read those genes and use that DNA, it's there. Do you remember how many chromosomes humans have? 23 pair, right? All right, so back to our animal cell. Here's our nucleus with the chromosomes inside containing the DNA. The nucleolus, which produces ribosomes. Make sure you can recognize all these cell parts. So let's go to ribosomes then, the protein factories. I remember ribosomes making protein because of the first three letters, rib. Ribs have a lot of protein, plus they're delicious. So that helps me remember that. Actually, ribosomes are made out of a type of RNA, a nucleic acid. Basically, the RNA is called ribosomal RNA, and it's instead of being in a straight line like the RNA in your nucleus, it's clumped in two clumps, actually. You'll see there's a top clump and a bottom clump. Perfect for when you have a string of RNA that has just copied a gene from DNA in the nucleus and has come out to the ER to find a ribosome. Perfect for clumping and attaching on, like right around that string of RNA. Remember that RNA is called mRNA or messenger RNA for short because it's carrying the information from a gene in DNA. So ribosomes then can exist on the ER or floating in the cytoplasm, and they clump around that mRNA to begin the primary structure of building a protein or a polypeptide. So look for little dots. Ribosomes are small. Here they are floating around in the cytoplasm, and here they are on the ER. And because all living things require proteins, even bacteria will have ribosomes. Bacteria won't have these other things like mitochondria and lysosomes, but they will have ribosomes. But remember, ribosomes are technically not organelles anyway. They're clumps of RNA. The ER, such a long word, endoplasmic reticulum. Endo meaning inside, plasm, inside the cytoplasm. These are basically membranes that form tubes. Kind of like, I think of like ER as like a lot of hallways. I get lost in the ER all the time. And so it reminds me, the ER, endoplasmic reticulum, that way. Under an electron microscope, here's a picture at the bottom. It looks like little tubes, and then you see the little dots on there. Those are the ribosomes. So that's why we call this the rough ER, when you see little dots or ribosomes on the ER. These are technically right outside of the nucleus because that's where we're going to build proteins. Oops, there's also smooth ER, which does not have any ribosomes on it. Smooth ER helps to make more membrane material and some other things as well. So there's our ER highlighted in red in our cell picture. As our cell picture is growing, you'll notice this is right outside the nucleus, but we have something very similar looking that's a little further out. I bet that's next. Yep, there it is. The Golgi apparatus, sometimes called the Golgi bodies. Basically, it functions to finish up the proteins, finish that 3D shape and get all the polypeptides attached in the right folds, and even vesicle it off to maybe send to a different part of the cell or send into the bloodstream. So it's like the shipping department, like UPS. And there it is in the picture. You'll find that further out from the ER, but it looks a lot like the ER. But both of them are basically tubes of membranes that are really good at vesicling off or bubbling off. All right, I hope you remember this. I love this picture. I love to go over this as much as I can. So you've got the DNA there in the nucleus. That's where the recipes for making proteins are, the genes, about 25,000 to 30,000 genes. It's stuck in the nucleus because it's such a big molecule, so it unzips in the area of the gene. RNA gets in there, makes a copy of that. Now it's called an mRNA, and it'll transfer that information through the nucleus out to the ER, find a ribosome. The ribosome, made out of RNA, clamps around that mRNA and starts stringing together the right amino acids, which is the primary structure of a protein, or making a polypeptide chain. Then we can vesicle that off from the ER and send it to the Golgi, the neighbor nearby, where we can finish the folds, finish adding other polypeptides to it, vesicle it off again, and send it to where it's got to go. Golgi is like the shipping department. I love that picture. So also, we can't ignore the plant cell. So there's all those things we just looked at in the plant cell as well. So looks a lot like the animal cell, just has that kind of main structure to it, a little less flexible with that cell wall, and then the chloroplast making green color, and the big old central vacuole for storage of all that sugar that those plant cells are making. Remember, both plant cells and animal cells have mitochondria. So we have to make ATP. All right, job number 3, cells need to make more cells. That's reproduction. You're a multi-celled organism. You've got to replace what you've got. So our cells make more of themselves by a process called mitosis, a process we'll look at more closely in a chapter that will come sooner than we think. The organelles that help with this are the nucleus, because it houses the DNA, and the centrioles. Centrioles are made out of protein. Whoa, there's protein again. That look like cool little star spirals like this. So these are centrioles. When you have 2 centrioles, a pair of them, that whole area with even the little spikes going out from it, which are also little proteins, that whole thing is then called a centrosome. So one cell has a pair of centrioles or one centrosome, the whole thing. This helps to coordinate cell division. This is where what's called the mitotic spindle will come from. The proteins that will kind of come out from this like a spider web to grab chromosomes and pull them apart. So this is what controls that. And there's one pair in each cell. When it's time to divide, though, that pair will actually multiply into 2 pair so that they can go to opposite sides of the cell and then help pull that cell apart. So there we have it, our 3 main jobs. Making energy, making proteins, and making more cells. our introduction to cells. The information I'm going to talk about now is on that third PowerPoint or Google Slides that I've included on your Google Classroom. A lot of it is, there's some repeats of what I've already done, some review, but I want to talk a little bit more about some other parts, like the cytoskeleton. When I think cytoskeleton, this is what I think. It's like the scaffolding for the cell. Look at that. Guess what cytoskeleton is made out of? If you said protein, you're right. So the structural support maintains the cell's shape. It anchors the organelles. It also is involved in movement. So if a cell has cilia or flagella, that's connected to the cytoskeleton, which has little contractile proteins that can slide past one another, actin and myosin, so that it can move. And it's also involved with regulation by organizing cell structures and activities of the cell. When you use an electron microscope to look at a cytoskeleton, this is what you see. You see kind of larger protein filaments, and you see smaller ones. So all of those are divided into different categories depending on their structure and how big they are. Some are called microfilaments, some are intermediate filaments, and some are microtubules. And then if I look at more of an animated picture, this is what it looks like, and it really helps me see how those different organelles are anchored in the cell. I will have a little side note that mitochondria are actually able to move within that cell, but all these other ones are really anchored down. Oops. And then we already talked about the centrioles, which are made out of very similar material, similar proteins as the cytoskeleton. Remember, there's a pair of centrioles that organize microtubules into what are called spindle fibers, what we know of as the mitotic spindle, to help with mitosis. So we see that there, and we can see the mitotic spindle filled with tons of little spiderweb-looking proteins here in this electron microscope picture. All right, then you also have some slides that come from, it's number 2. And a lot of that is review that I've already talked about. However, I want to bring your note to just a few things. The word apoptosis. All right, this is basically cell suicide, programmed destruction of cells in multicellular organisms. If you have a bad cell, you don't want that cell hanging out and causing problems or becoming cancerous. You want to get rid of that cell. So cells have signals to tell themselves when they gotta go. And if that works, then it'll help us avoid other problems like cancer later on. We'll talk more about apoptosis when we talk about mitosis in a later chapter. All right, this slideshow also really focuses on the mitochondria and chloroplasts a little bit more, but we'll focus on those in chapters 9 and 10 when we talk about cell respiration and photosynthesis. So I won't spend extra time on them right now. However, I do want to bring up some interesting things about them rather than like their functions within the cell, but some interesting things about them. Mitochondria and chloroplasts can reproduce on their own or divide on their own, and it looks a lot like a bacteria when it's dividing and reproducing. So, almost all eukaryotic cells have mitochondria. There may be one very large mitochondria or hundreds to thousands. And let's see here, chloroplasts. Yep, they can divide on their own as well. I'll skip through a bunch of that. And just to mention the fact that mitochondria and chloroplasts are very different and special. They're not part of the endomembrane system. They're not connected to the ER or anything like that. They can move around on their own if they want. They grow and reproduce on their own. They actually make their own proteins. It's like, hey, I don't trust the cell to make my own protein or to make my proteins for me. I'm going to make my own. Well, to make your own proteins, you need a recipe, right? So they actually have their own DNA. And get this, their DNA is circular in a ring, which actually resembles bacterial DNA. Bacteria have a single chromosome that's in a circle, DNA connected as a circle. So it's called a circular chromosome. So their own ribosomes, making their own proteins with their own DNA, moving around by themselves, reproducing when they want to. What does that remind you of? If you say bacteria or prokaryotes, you would be right. So there is a theory called endosymbiosis theory, because we wonder, how did eukaryotic cells come to be? Because it seems easier just to make prokaryotic cells, which is why they were first in the list of life. But how did we get those eukaryotic cells? So endosymbiosis theory helps to explain that, thanks to Lynn Margulis, the University of Amherst. And so basically how that works is that over time, the membrane started folding in on itself, as we see in the picture here, just infolding into the cell, into the cytoplasm. If it infolds enough, it could enclose the middle section there and make a nucleus and kind of disembark from the rest, make its own little organelle, therefore having a nucleus. That's on our road to making a eukaryotic organism. And then the ER, I can see that right there, which is basically just membrane tubes. And then possibly if a cell engulfed a bacteria who made ATP for the cell, perhaps it didn't digest it and then just kept it. And of course that mitochondria would be able to reproduce within the cell and became a permanent inhabitant. And the same thing with chloroplasts. If a cell engulfed a photosynthetic bacteria with chloroplasts making sugar, wouldn't you want to keep that too? Something making sugar for you so you don't have to go out and find your own or eat your own or absorb your own. So this is called endosymbiotic theory. And there's tons of evidence supporting that, especially with the structure of mitochondria and chloroplasts, to help us explain how we got eukaryotic cells, going from a prokaryote to a eukaryote. We'll look at this a lot more when we get into chapters 25 and 26 as well, but we should start to understand it now and appreciate the differences of prokaryotes versus eukaryotes and the differences between mitochondria and chloroplasts and other cellular organelles. And don't forget those equations, right? Photosynthesis and respiration. Photosynthesis, carbon dioxide, water, and sunlight gives us glucose and oxygen. Respiration, we've got glucose and oxygen giving us carbon dioxide, water, and of course, ATP, done in the powerhouse of the cell, right? Mighty mitochondria. All right.